psilocybin is found in a number of different mushrooms that, when ingested, can cause hallucinogenic effects. When ingested, psilocybin is metabolized by the liver into psilocin. Psilocin is a prodrug, which means that it is inactive until activated. It is the psilocin's activity at a subtype of serotonin receptor known as 5-HT2A that causes effects. When psilocin binds to 5-HT2A, it only causes a small reaction in comparison to the natural reaction that serotonin causes when binded to 5-HT2A. While psilocin mostly interacts with serotonin receptors, it also sometimes interacts with subtypes of histamine, dopamine, and adrenergic receptors. Like a lot of other substances, repeated use of psilocin will build a resistance. Interestingly, if someone builds a tolerance for LSD, they will also build a tolerance for psilocin, and vice versa.
The 5-HT2A receptor is a G-protein coupled receptor that receives the ligand psilocin. When this receptor is activated, the GQ protein activates the cell signaling pathways. After reception, the G-protein coupled receptor phosphorylates GDP into GTP. From there, the G-protein can then activate either phospholipase A2 or phospholipase C. For this video, we will be focusing on the phospholipase A2 pathway because that is more common for psilocybin. Phospholipase A2 then synthesizes diaglycerol, which then activates the enzyme protein kinase C. From the protein kinases, IP3 accumulates in the cytosol, causing calcium to be released from the endoplasmic reticulum. The calcium being released in the synapse changes the charge of the cell, which allows the neuron to fire at different rates. Most of the 5-HT2A receptors are found in the cerebral cortex and olfactory tubercle, which is why they produce a physiological response. Some reactions include, but are not limited to, neurological excitation, vasoconstriction, and smooth muscle contractions.